Look inside this room. The furniture, the floor, the rug, everything in sight is made up of chemicals. In fact, so are we. The tissues, cells, and fluids of our bodies are all composed of different chemicals. Yet, in important ways, these chemicals are different from those in a curtain or a chair. For one thing, the chemical reactions in a chair, if they take place at all, occur so slowly that years from now the chair will probably look the same. The chemicals in the human body, on the other hand, undergo billions of reactions every second. How does this happen? Human beings, along with all living things, have enzymes that facilitate chemical reactions. In fact, if it were not for enzymes, we wouldn't have the muscular energy to turn a page, or the nervous energy to read a sentence. Indeed, without enzymes, life itself would be impossible.
Let us take another look at the chair. What prevents the molecules in a chair from reacting? The answer is that these molecules need to be pushed into reacting. The molecules of an inanimate object, such as a chair, face a situation similar to that of a skier at the bottom of a steep hill. In order to have a good downhill coast, she'll first have to spend a great deal of energy getting to the top of the hill. Once at the crest, she can relax and look forward to a long, almost effortless downhill ride. Similarly, in order to react chemically, molecules must first be provided with some energy to become activated. In a way, they must surmount an energy hill, known as the energy of activation. This hill can be thought of as a barrier. Once at the top, the molecule has crossed the barrier and is free to react and form different molecules called products. But how can molecules be brought to these energy peaks where they're capable of reacting? One way is to heat the molecules up. Heat causes molecules to move around faster and bump into each other. And the harder and more frequently molecules collide, the more they react. When molecules react, chemical links called bonds are either formed or broken. By applying intense heat to the wood in a chair, for example, we would cause molecules to speed around, break apart, and combine with oxygen in the air. In other words, the chair would burn. But such intense heat would have disastrous effects inside the body. It would kill the body's cells, and life would be impossible. Another way to increase the likelihood that chemicals will react is to increase the concentration of the chemicals involved in the reaction. The concentrated supply of oxygen in wind and rain, for example, causes certain metals to oxidize or rust. But again, this is not how living organisms work. A third way to facilitate chemical reactions is with catalysts. A catalyst is a special substance that speeds up a chemical reaction without itself being affected. In the human body, catalysts, rather than heat or concentration, are used to speed up chemical reactions. Catalysts that do their work in living organisms are called enzymes. Like all catalysts, enzymes work by increasing the likelihood that chemicals will react. It's as if they lower the energy hill chemicals must climb before they can react. An enzyme can't bring about a reaction that would be otherwise impossible. All an enzyme does is make it easier for reactions to take place.
An enzyme is a type of protein. Proteins are large molecules made up of chains of chemicals called amino acids. The sequence of amino acids in these protein chains determines what protein it is. The largest enzymes are made up of many long interwoven chains containing thousands of amino acids. these chemical chains bend and fold into characteristic shapes. An important characteristic of each kind of enzyme is its unique architecture resulting from the pattern of these folding protein chains. The substances enzymes act on are called substrates. The unique shape of an enzyme allows it to fit closely with a particular kind of substrate, somewhat in the fashion of pieces of a jigsaw puzzle. When the substrate and enzyme combine temporarily, they form an enzyme-substrate complex. The substrate joins the enzyme at a particular place on the enzyme surface called the catalytic site, or active site. The shape of this site and the chemical nature of the amino acids in the site determine which substrate is welcome and which ones are not. The enzyme lowers the amount of energy needed to start a chemical reaction. This reaction leads to the liberating of substances called products. After the products are liberated, the enzyme is free to attach to another substrate. The fit between a substrate and an enzyme is so precise that most enzymes can distinguish among the thousands of compounds in the body and combine with only one particular substrate. Stated another way, for any enzyme, there's usually only one substrate that will fit in its catalytic site.
There are over 1,000 kinds of enzymes at work in the human body. In fact, there is generally one kind for each different chemical reaction that takes place in the body. Some enzymes are specific as to where in the body they do their work. For example, enzymes that help our nerves to transmit impulses or that enable our eyes to see work within nerve or receptor cells. Enzymes that catalyze reactions that enable muscles to contract are found in muscle cells. Some enzymes, however, are found in all the body's cells. Some of these enzymes, for example, control the reactions that cause life-supporting energy. There are also enzymes, such as those that help digest food, that do not operate within cells at all, but are part of the fluids secreted by the body's glands.
Let's examine more closely how enzymes secreted by different parts of the digestive system operate. Digestion is a multi-step process through which the proteins, carbohydrates, and fats in our diet are broken down into simpler compounds that can be used by the body's cells. It is a process that can begin in the mouth the moment we bite into a piece of food. Glands in the mouth secrete saliva, which contains the enzyme salivary amylase. This enzyme begins the process of breaking down starches, a kind of carbohydrate, in foods like pasta and bread. Other aspects of digestion take place further down the digestive tract in the stomach. Here, for instance, the enzyme pepsin begins the job of breaking down the proteins in such foods as cheese, meat, and fish. After the stomach, food particles travel to the small intestine, a 20-foot tube coiled in the abdomen. A nearby gland, the pancreas, secretes a number of enzyme-containing fluids into the small intestine to aid in digestion. For example, the enzyme pancreatic amylase continues the process of breaking down starches. The enzymes trypsin and chymotrypsin take particles of protein partly digested by pepsin and break them down further. And fats are broken down by enzymes called lipases. Altogether, there are dozens of different enzymes involved in digestion, a few of which are shown on the screen. Each one of these enzymes performs a specific task. The ability of enzymes to break down compounds in food has been put to a number of commercial uses. For example, cheesemakers use rennin, an enzyme from the stomach of a calf, to curdle milk. The enzyme papain in the papaya fruit can split proteins, so it's used as the active ingredient in commercial meat tenderizers. And some detergents contain enzymes that help remove stains by breaking